In this video, I am going to start or introduce biology. I have selected one chapter from a standard 10 class, and I have selected chapter Heredity and Evolution. This is one of the most important chapters, and it is highly confusing in nature also. Because before going to start these chapters, we need to discuss some topics, some terms that is related with these chapters. Then only this chapter will be clear to you. So, the chapter I am going to start is heredity. So, before going to start this chapter, what are the important points, what are the important terms that we have to study, we have to know? First is the types of reproduction. Second is the types of cell division. Third part is what is heredity? Then what is variation? Then what is chromosome? Then what is gene? What is alleles? Then it is homozygous gene. Then it is heterozygous. Then genotype. Then phenotype. So these are the terms you need to study, you need to understand, then it will be easy for you. Very easily you can understand this chapter. So first is the types of reproduction in plants and animals. You can use animals only here, plants also. So there are two types of reproduction. First is the asexual and second is the sexual reproduction. In the lower organism of the plants, asexual reproduction takes place, whereas in higher organism of the plants and animals, sexual reproduction takes place. So here asexual reproduction means formation of organism or individual without fusion of gametes. Without fusion of gametes. Here, sexual reproduction is just opposite of that. That is formation of organism with fusion of gametes. In human beings and other, there are two types of gametes. Gametes produced by father cell is called sperm, or gametes produced by mother cell is called egg. Here one more information, haploid and diploid. I give haploid and diploid. Haploid cell and diploid cell. Second difference is asexual reproduction is that the offspring is carbon copy. The offspring is the carbon copy of the parent, It is very difficult. Hardly you can distinguish which one is parent and which one is offspring. But here, offspring is not carbon copy. Not carbon copy. There is a differences between parents and their offspring. We cannot say that father and child are carbon copy of each other, mother and child are carbon copy of each other. There are some differences. So the differences present in the organism of the same species is called variation. So here I will discuss what is the variation, what is the heredity. First type of reproduction, asexual and sexual. So here when we are going to use the concept of heredity, then this is applicable only for sexual reproduction. Heredity concept we don't use in asexual reproduction because here the carbon copies, parents are, offsprings are carbon copy of their parents. Second is the types of cell division. There are two types of cell division, very important information, and that you have to know before going to start this chapter. First is the mitosis and second is the meiosis. In mitosis, a parent cell gives two daughter cells and the chromosomes of the parent cell and daughter cells are same. 2n. This 2n chromosome is called diploid. This is diploid chromosome. Whereas in meiosis, a mother cell produces 4 daughter cells and the chromosomes of the daughter cell, mother is diploid and offspring daughter cells are haploid. The daughter cells are, we can use the word daughter cell. Here also two daughter cells or two cells, chromosome numbers are same, so daughter cells are haploid. N chromosomes are called haploid. So what do you mean by haploid? N number of chromosomes. What do you mean by diploid? 2n number of chromosomes. For example, if human beings has 46 chromosomes, then it is a diploid. For n chromosome, it is 23. So this is the point. Now what is heredity? You can say that transmission of genetically based character from one generation to another generation is called heredity, or transfer of genetically based character from one generation to another generation. So you can see that transfer of genetically based character from one generation to another generation. Variation, what is variation? Differences among individual of same race is called, or same tribe is called variation. What is chromosome? You can say chromosome is the heredity character. So actually what is going to be transferred from parent to their offspring? It is the chromosome that is going to be transferred from parents to their offspring, and that chromosomes are made up of genes. You can say that chromosomes are made up of genes, or you can say that chromosomes are made up of DNA, that you will study in higher classes, the composition structure of DNA. Alleles, in a gene, a pair of character, contrasting pair of character, contrasting pair, opposite pair, contrasting pair of character. That is called alleles. For example, if we take tall plant, then tall plant can be represented like capital T, small t, capital T, small t. When the two alleles are same nature, it is called homozygous. This is also homozygous, and when the two alleles, one pair of contrasting character are different, it is called heterozygous. Very important information here than homozygous, heterozygous. What is genotypes? If we say, what is this? You can say this is a tall plant. But this is also tall plant, this is also tall plants, but you can say this is homozygous tall plant. So if we see the external form, phenotype is the external form. So if we see the external form, all are the tall plant. Externally we can say all the three are tall plant. But if we see the genotype, genetic constitution, then we can say two plants are homozygous tall and one plant is heterozygous tall. That is the genetic constitution of plant is called genotype. So these are the information that are required to start or to know Gregor John Mendel experiment, that is law of dominance or different types of law that is required. So first I will going to explain Mendel's experiment. First I will explain Mendel's experiment. He used pea plant for the experiment. He selected pea plants because it is an annual plant. Annually its life is controlled and it has 7 pair of contrasting character like height of the plant, tall or dwarf, position, axial or plant terminal, shape of the pod, round or wrinkle, color of the pod, green or yellow, color of the flower. So he got many characters that was required for the experiment. All characters were present in this plant. That's why he selected pea plant. And what he did, he took 2 pea plant. One is the pure tall and another is the pure dwarf. Both are homozygous. And he observed that here after gametogenesis, gametes chromosome gets haploid. You can say 2n chromosome. Very important information. In a plant, 2n chromosome. In human beings, 2n chromosomes. But the gametes, either in terms of plants or in terms of animals, like sperm has n number of chromosomes, egg has n number of chromosomes. So here the number of chromosome is n chromosome, that is haploid chromosome. So in the F1 generation, first filial generation, F1 generation, he observed that all plants were tall. Very important information he got. He took tall plant also, he took dwarf plant also, he made a cross between both the plants and he observed that only the plants, all the plants were tall. On the basis of this, he gave his first law, that is law of dominance. Very important law of dominance. That if in a given organism or if in a given alleles, one alleles are expressing itself without being affected by other alleles, or you can say if one allele is presenting itself without being affected by other allele, then this is called dominant alleles. The allele which express itself, the gene that express itself is dominant character, and the character which doesn't express itself is recessive character. So he said is the tall is the dominant character and dwarf is the recessive character. So the character which express is the dominant character and which does not express is the recessive character. After that, for F2 generation, he produces he made gametes. For example, there are tall and dwarf, both the characters are there. This is 2n chromosome, 2n chromosomes. During the gametes formation, chromosome gets haploid, it gets half. So if suppose this is the plant, we can take male plant or female plant separately. So female will produce two types of gamete, capital T and small t, and male will also produce two types of gametes, capital T and small t. And on the basis of this experiment, he observed that this is the information he got. He got phenotype ratio, phenotypic ratio, very, very important information. Phenotypic ratio, that is observable character. This is tall, this is also tall, this is tall, and this is dwarf. So he said 3 plants were tall and 1 plants were dwarf. Or you can say 75% tall and 25% dwarf. So this is very, very important information you will have in the board exam. You can get question from this part, that what was the result of F2 generation phenotypic ratio. So remember, 3 plants were tall and 1 plants were dwarf. What is genotypic ratio? So genotype. Genotypic ratio, it is here, this is homozygous, this is homozygous, these are heterozygous. So you can say that 1 plants were tall, 1 plant, 2 plants were heterozygous tall, and 1 plant was homozygous dwarf. This is homozygous tall, genetic constitution, this is heterozygous tall, and this is homozygous dwarf. So genotype was 1:2:1 ratio and phenotype ratio was 3:1. So on the basis of this experiment, he gave 3 laws. First was the law of dominance. Second law is the law of segregation. Laws of segregation. And third is the law of independent assortment. So you can see that what is law of dominance? So we can get law of dominance on the basis of F1 experiment. Result of the F1 experiment say that in a given plant there are one pair of contrasting character. The character which express itself is the dominant character and the character which does not express is a recessive character. What is law of segregation? During the gametogenesis, gametes separated independently without affecting the other, and independent assortment, when one's gametes are separated, they fuse with other gametes randomly, independently, not affected by the other. So on the basis of these 3 laws, Mendel explained monohybrid experiment. This is very, very important information. Why it is called monohybrid experiment? Because he took one pair of character, tall plant and dwarf plant. Why it is called phenotypic ratio? Because we are getting observable character. Why it is called genotypic ratio? Because we are getting genetic constitution of the plants. Why it is called 2n chromosome? Because here diploid chromosome. Why it is called n chromosome? Because the chromosome numbers haploid. And when does 2n turns to n? This is the process of gametogenesis. Gametogenesis, 2n chromosome gets turns to n chromosome. So these all information about monohybrid experiment. Now, what is dihybrid experiment and what is its result, that I will explain. In his hybrid experiment, he took two pair of characters. He selected plants with two pair of character. First is the round and yellow, and second is the wrinkled and green. So in this he selected pure round, capital I, or pure Y, capital yellow, wrinkled, small r, or green. This is two pure plant, and its gametes RY and its gametes RY. In F1 generation, all plants were round and yellow. Now, in F2 generation, see, two characters are there, so four types of gametes. Very, very important. One character, two types of gametes. Two character, four types of gametes. So you can say round yellow, round green, wrinkled yellow, wrinkled green. Here also round yellow, round and green. Very, very important. Be careful while making cross. This is very, very important. So first is RRYY. This one is RR, capital Y, small I. This is capital R, small r. This is both R. This is capital. This is capital. Then this capital is small. This is capital. This is small. Here R, capital Y, small I. Here R, capital Y. Here R, capital Y, small y. Mind it, here R, both are small y. Here capital R, small r. One is small, one is small. One is here capital R, small r. Both are small y. Here both, one is capital, one is small. One is capital Y and one is small y. Here both are capital. Here this one is the last. Y. This is small. This is small. Both are small. This is Y. This is RR. See, on the F1, F2 result, he got the result 9331. That is 9 plants were round and yellow. 3 plants were wrinkled yellow. then round yellow, round green. You can write round yellow. Then 3 are round green. Then wrinkled yellow. And this one is wrinkled green. So this is, if you convert this is round yellow. This is round yellow. So if you write this, you get information like in terms of 9331. So this information is very, very important, that what was the result of F1 hybrid, F1 monohybrid experiment, F2 monohybrid experiment, what was the result of F2 dihybrid experiment result, 9331. This is the most important information related with Mendel's experiment. So there are some questions we have to discuss. First question is, how do Mendel's experiments show that traits may be dominant or recessive? You can say that F1 result of monohybrid, he got all the plants were tall, although there was dwarf character also. So he gave that the character that express itself fully is dominant character, and the character which does not express is the recessive character. Second is, how is the sex of a child determined in human beings? Very, very important information. Sex determination in the plants, that I will discuss. How will sex determine in a plant? So here first we have to know chromosomes, types of chromosomes. To know the process of sex determination, we need to know types of chromosomes. This is most important part, very, very important. Why? Because this is the part of our life. Some childs are female childs, some childs are male childs. So what are the factors which affect itself? Here, first we need to know chromosomes. There are two types of chromosomes. Chromosomes are the hereditary factor, and there are two types of chromosomes. First is called autosome, and human being its number is 44, 2n, and sex chromosome, its number is XX in female, that is 2 chromosome. Altogether 46 chromosomes are present in human beings. 44s are autosome and 2 chromosomes are sex chromosome. So in the female, both the chromosomes are XX, whereas in male, you can say, father say, 2 different types of chromosomes are there, X and Y. Mother always produces a single types of gamete, that is homogametic in nature. Very, very important. Very, very important. Homogametic. Mother is homogametic in nature, whereas father is heterogametic. 2 types of gametes are produced in the father. So we can say father is heterogametic. And the born baby is female depends when X chromosome of mother fuses with the X chromosome of the father. The baby is female baby. Whereas when the X chromosome of father fuses with the Y chromosome of X chromosome of mother fuses with Y chromosome of the father, then it gives male child. So you can say the born baby is whether male or female. It depends upon chromosome of the father, not upon chromosome of the mother, because mother is homogametic in nature. She has only one chromosome, that is X chromosome, whereas father has two different types of chromosome, X chromosome and Y chromosome. And law of independent disturbance, fusion of gametes occur independently. So there is no any rules that which type of gametes will fuse. It may be XX or it may be XY. So in this way, sex of a child can be determined. Here one information is related with blood group also. In CBSE book NCERT it is given. So I want to explain this part also, ABO blood group. In short, I want to explain this as a trick so that you will be able to solve this question. The question first, let me give some information. After that, I will discuss this question. Human has three different types of blood group. Four different types of blood group: A, B, AB, and O. There are four different types of blood groups are there in human beings. So just I'm giving you short tricks so that you can understand. I'm not giving the entire concepts. Write A blood group like this: IA, IO. Even though A blood group is dominating over O, B blood group is dominating over O, you must know O is the universal donor, whereas AB is the universal acceptor. So you remember A is dominating over O. If gene of A and O both are present, then blood group will be A. So B we represent like IB, IO, AB will represent IA, IB, and O, IO. Just this information I'm going to give you. I'm not going to details because not much information about this part is given. So your question is a man with blood group A. So the blood group of father is A. And woman, that is mother, blood group is O. So you can write A blood group as IA, IO, and O blood group I as IO, IO. Here, their daughters has blood group O. Is this, no, here question is a man blood group A marries a woman with blood group O. So first question is, what is the blood group of their children? So just you separate it's this part IA, IO, this only single gametes, single part IO. So here, what type of children? That's parents. They will have two type of children, either blood group A or blood group O. So in this way, we can find blood group of the offspring, or we can guess also. Suppose father has A blood group. We write this like this: father has A blood group and mother has B blood group. So A is IA and IO, B is IB, IO. Now, here AB, here IA, IO, here IB, IO, and here, mind it, all type of blood group can be present in their children. You can say 25% children will be AB blood group, then 25 A blood group, then 25 B blood group, and 25 O. All type of children will have all type of blood group. It may be AB also, it may be A also, B also, or O. Here, if father is A or mother is O, then only two types of blood group will present, either A or O. No AB blood group. Similarly, if father is B and mother is O, then again here only B and O. So in this way, we can find the blood group of the children, or we can guess what type of blood groups may present in their children on the basis of blood group of father and mother. Then after that, there is a question, NCERT book it is given, how is the equal genetic contribution of male and female parents inserted in the progeny? Equal genetic contribution of male and female parents inserted in the progeny. So you can say that a progeny or offspring is the result of fusion of male gametes of the father and female gametes, or you can say mother gametes and father gametes, and the number of chromosome of father and mother gametes are haploid, equal contribution. So you can say 23 chromosome, that is 22 plus X. Mind it, 22, suppose the child is male child. So you can say 22 plus X of mother and 22 plus Y of the father. Suppose this gametes fuse, then the child will be a male child. So here equal gametes, equal contribution and chromosomes are fused to produce a new generation. So we can say that equal genetic contribution of male and female parents inserted in the progeny. So in the next video, I will take some other topics related with the biology. This chapter is little bit confusing, and all terms are related with each other. That's why first I have separated all the terms. I have explained all the terms first, and then I started the experiment, monohybrid experiment and dihybrid experiment. So I hope you will understand this topic, and in the next video I will bring some other concept. Thank you.